is going to be How Networks Change with Time by Yongjin Park and Joel Bader, and Yongjin Park will give the talk.
Okay, thank you. Thank you for coming, and thank you for inviting me for the ISMB meeting. The title of the talk is How Networks Change with Time. I am Yongjin Park, and this is joint work with my advisor, Joel Bader. Here's the email address if you have any questions or comments, and I would love to discuss. If you want to study some biological systems, for example, here is a chemostat or bioreactor, you want to measure what's going on inside of this system in this time series. So you can measure mRNA, you can measure proteins or interactions and complexes. And what's actually characterizing in the biological state of this system is we can get out from the mRNA fluctuation, because mRNA is actually most widely used high throughput experiment that we're using. But other data sets like protein level measurement or protein-protein interaction measurement or protein complex measurement, those are actually difficult to access. What's going on inside this system, to measure the activity of this system, we will actually have to get the proteins and their other macromolecules and their interactions. Okay, this setups our problem of dynamic network clustering. We want the time series experiment, and using the mRNA as a time series proxy to the protein levels, and we assume it's a steady state model. We're actually looking at yeast as a model organism, so the expression is actually more straightforward. And the protein-protein interaction, we pull out the public database from Interactome DB, for example, BioGrid, and then our problem is how given the dynamically changing interactions over the time, what is the complex that are changing over time? So input is dynamic networks of G, I denote the network data as G, and from G1 to GT, and the output is time evolving network complexes. The open question is how, when we look at this dynamic complexes, is there time correlated assembly or disassembly of this complex? The data set we are looking at, motivating example was metabolic cycle, and which was published in the Science by Tu et al. And they're looking at the chemostat, and they start from the batch culture of these yeast cells, and then suddenly the yeast cells grow, and then the oxygen level in the chemostat actually drops down to the 0%. And then if you cut down the nutrition source, then the oxygen levels go up, and then cells don't grow much. And then you add another round of food, add the glucose, and then you see the nice oscillating pattern of the yeast metabolic cycle. But in the Tu et al. Paper, they characterize three specific phases of yeast metabolic cycle. One is the oxidative, denoted by ox in color red, and reductive building, which is colored by green, and reductive charging phase. They studied the yeast metabolic cycle using the microarray, and it takes about 3 hours per metabolic cycle, and about half of whole transcriptome is periodic, and three major groups are OX and RB and RC. During the OX phase, cells generate energy and ribosome synthesis and amino acid synthesis, and the reductive building phase, cell divide, and reductive charging phase, there's lipid metabolism and glycolysis. And they proposed an interesting hypothesis, which is temporal compartmentalization, and which actually separates three groups of genes by time trace. And here's data processing. Given this mRNA arrays, the x-axis is time and y-axis is genes. We map the interaction patterns to the BioGrid interactome, and looking at we also focused on previously identified periodic genes and their pairs, and we removed the anti-veg edges, which is, for example, if you want to look at the interaction at the RV phase, you don't want to include the pairs of interaction that are active. One end of the edge is active at the OX phase, the other end of edge is active at the RC phase. This is antiphasic, so we remove those antiphasic edges. And we generated 36 network data, which consists of 3 cycles and 12 networks per cycle. Here's the problem. We have snapshots of networks. And what is the model that are evolving over time that actually describes dynamic networks that are changing over time? So first, let me break down this modeling question as two parts. The first part is how do we model each single snapshot, and then how do we connect these different snapshots, transitions. And we use the kind of stochastic block model. Stochastic block model is generative model. It generates adjacency matrix, which is network, given group structures and parameters, block parameters. Usually block parameter is very much smaller than the adjacency matrix. Adjacency matrix sits on the O V square. V is number of vertices, and block parameters usually O K squares, and the K is number of groups. Most of the interacting data is just binary, just 0 or 1. So we use the Bernoulli trial as a likelihood model. And here's a comparison of different complexities. So networks, we have O V square, and flat block model, we have O K squares. But another class of model we're considering is how about tightening down this group together some of the parameters that are similar to each other, which we use as a hierarchical model. Hierarchical model was presented by Closer, Moore, Newman, Nature paper, and we extended their paper, their model to biological problems. Here's an overview of a clustering method. Given this, using these networks, O V square, we first using the agglomerative clustering, and then we make the fully branching hierarchical model, which is an OV, and then we collapse some of the subtrees, then we can get the more compact representation of hierarchical model. The other way you can get to the compact hierarchical model, you can first build a flat stochastic block model, and then you summarize by hierarchical model, but we're going the other way. And the method actually consists of 2 parts. The first part is building the fully branching hierarchical model. We do merging of the pairs, and then we do collapsing in the end. First, let's look at the merging. It builds a maximum likelihood guide tree, and start from each vertex, each network gene or proteins as one group, each group, and then we start merging what's the most likely pairs that we're trying to get iteratively. So hierarchical block model. And then we do collapsing. We use, by the merging order, we get the nice spectrum of a model comparison. So starting from the fully matrix itself and then get to the fully branching hierarchical model, we get the nice spectrum of model comparison. And then following this merger orders, we measure the base vectors. So base vector actually marginalize the parameters. So we try to avoid the overfitting to the data set. So this actually gives us a nice automatic stopping criteria. And then we can do global model comparison score. Yes. Then finally, we get the much more smaller and more reduced model complexity of OK. And now we have a good method for single snapshot clustering. And how do we connect different snapshots? So to connect different snapshots, we assume it's smooth and slow transition of the models. So we use the idea of Ericsson's bioinformatics paper, and they use kernelized merging and collapsing score. And the kernelized score is averaging of time averaging of different model comparison scores. For example, left one is maximum likely guide tree building score, and the other score is collapsing score. We starting, for example, if you're looking at time t and you want to consider all the other neighboring snapshots, if you're farther from this time t, then you probably don't consider that much. So using the RVF coronal weights at time t, we get the nice smoothing, and also we can represent higher order dependencies. But next stage is we get the nice bottom level clustering and the collapsing. We have nice hierarchical model and clusters. But the thing is, voluntary clustering doesn't really say much about labelings. So the first snapshots clusters does not necessarily match to the next snapshots. So we have to match them across different snapshots. And also some proteins actually change in memberships. So, or some groups may break down and then merge together. That can actually happen during the dynamic networks. But what we had actually observed in our data set was just membership switch. And the other piece we used was matchem algorithm, which is basically EM algorithm. So during the Yeast step, you guess what's the possible label. Here, I would say it's a color. And we guess the colors by loopy belief propagation. And during the M step, we estimate the parameters by maximum likelihood, and then we keep going on E step and M step again. And this graph actually shows a vector graph representation of our matching problem. And this actually by this matching algorithm, we can handle discontinuity of the clusters. But if you're using bipartite matching, then you'll not be able to catch those things. Here's a result. We applied our result in the Yeast metabolic cycles of the periodic genes. I grouped them, those dynamic clusters that are changing over time. So x-axis is time, and grouping was just basically by their gene expression profiles, and OX and RV and RC. And the colors represent the normalized expression levels, and cluster size was proportional to the actually square size. During the OX phase, we pull out the genes that are corresponding to OX phase and their clusters. And the keywords of the gene description was ribosomal and polymerase or kinase. Basically, the RNA, DNA polymerase and ribosomal. And during the reductive building phase, it's more focused on the mitochondrial ribosome. And reductive charging is more diverse function. And what specific local views we're interested in. The first local view was a mitochondrial ribosome complex. And most of the genes in the complex, they're responsive to RV phase. They're highly active during the RV phase. But one gene actually sticked out was RSM22. They're actually overexpressed before the other genes get overexpressed. So we were thinking that this could be some of the regulations going on by RSM22 and spreading out the signal to the other members of the complex. So it turned out that RSM22 was a SAM-methyltransferase, and it was also known that the ribosomal RNA complex was stabilized or assembled by the methylation of ribosomal 3' end of ribosomal RNA. So this actually proposed one of the interesting hypotheses from our clustering results. And the second example was nuclear pore complex. Nuclear pore complex was responsive to the OX phase. So during the oxidative phase, they're highly overexpressed. But the interesting part is there is consistent part across the snapshots. Those are nuclear pore proteins and caryophylline proteins. And nuclear pore proteins provide structures of the complex, nuclear pore complex, and caryophylline provides a mediator function. And another transient subnetworks that are only attached at the OX phase, which is ribosomal editing proteins, RRP proteins. And this actually suggests interesting hypothesis RNA processing could be coupled with the transport of RNA or DNA at the nuclear pore complex. Okay, here's a conclusion. We propose novel dynamic network clustering method. We can identify smoothly changing clusters. We use the Bayesian model comparison for cluster, and the related works of the Bayesian model comparison of the hierarchical model. It was, but their data set was just points in the space. So Heller and Garmani and Blondel and UIT, and the kernelization idea was inspired by Erickson's bioinformatics paper. And we also proposed novel dynamic set matching method, which enables us to track down temporally evolving clusters, and we use the expectation maximization and belief propagation. And maximum bipartite matching was also by belief propagation was also provided by other peoples. And by giving the results, we can hypothesize some of the new biological insights. One example was early expression of the stabilization proteins. So once it gets overexpressed first and then stabilize the remaining complex. And the other example was there's a transient recruitment of subclusters. We looked at the nuclear pore complex and RNA processing. And I would like to acknowledge funding from NIH and Clever Foundation. I would like to thank support from High Tech Biology Center at Johns Hopkins and Technology Center for Network and Pathways. Helpful scientific discussion with David Bader from Georgia Tech and Chris Moore, University of New Mexico. Special thanks for my family. This presentation was made possible by travel fellowship worked by ISCB with grant funds obtained from DOE Science Hub Office of Science. And here's the emails. You can contact me. I would love to discuss. Thank you.
Thank you. I had a sort of a two-part question. I guess I'm sorry if I missed something, but I wasn't clear whether the evolving network you were using was derived in some fashion from the mRNA data, or whether you actually had network like protein-protein interaction data that was temporal.
Oh yeah, it was overlaid, given the protein-protein interaction, the whole possible physical interactions in the biogrid.
That's static, that network
That's
is
static, and we use the dynamic mRNA profiles to be able to distinguish which edges are actually present at this time point, which edges are not present at this time point.
So I guess the second question would be, if you had dynamic data at both levels, would this method generalize to that case?
I'm sorry, I didn't hear you.
If you had dynamic mRNA data and dynamic interaction data, would this method generalize to that case?
Yes. Yes, that will help, yes. Because most of the probabilistic model is generative, so there's lots of rooms we can actually combine with other sorts of high-throughput sequencing, high-throughput data sets.
Thank you.